In 1952, one of biology's biggest questions, what a gene is actually made of, was answered with a kitchen blender. For decades, the smart money had been on protein. The smart money was wrong. By the 1950s, biologists knew where genes lived, on the chromosomes, coiled inside every cell. And a chromosome was made of two kinds of molecule twisted together, protein and DNA. One of them carried the instructions of life. The favorite, by far, was protein. Proteins are built from 20 different units, strung in any order, endless variety, enough to spell anything. DNA had only 4 units, thought to repeat in a dull, monotonous pattern, surely too simple to write a living thing.
There had been a clue, and it had been ignored. 8 years earlier, in 1944, Oswald Avery and his colleagues had purified the very substance that rewrites one strain of bacteria into another, and found it was DNA. But few were convinced. Perhaps a trace of protein riding along in the DNA was doing the real work. So the question stayed open. The gene. Protein or DNA?
2 scientists at Cold Spring Harbor set out to settle it. Alfred Hershey and Martha Chase. Their tool was a virus, bacteriophage T2, which hunts the gut bacterium E. coli. And a phage is almost nothing, a coat of protein wrapped around a thread of DNA. It lands on a cell, injects something inside, and minutes later the cell bursts open with 100 new viruses. Whatever it injects to copy itself, that is the gene. Find what goes in, and you find the answer. The phage has exactly 2 parts, so Hershey and Chase gave each part its own radioactive marker. The chemistry made it clean. Protein contains sulfur but no phosphorus. DNA contains phosphorus but no sulfur. So they grew one batch of phage with radioactive sulfur, sulfur 35, which stains only the protein coat, and another batch with radioactive phosphorus, phosphorus 32, which stains only the DNA. 2 parts, 2 labels. Now whatever entered a cell would announce itself. They let the labeled phage settle onto the bacteria and inject. Then came the trick that named the experiment. They poured the mixture into a Waring blender, an ordinary kitchen blender, and switched it on. The blades didn't harm the cells. They sheared the spent phage coats off the outside, snapping the empty shells free from the surface. Then they spun the tubes in a centrifuge. The heavy, infected bacteria, and whatever was now inside them, sank to the bottom as a pellet. The light, empty phage coats stayed up in the liquid above. Inside the cell, or left at the door, the 2 parts of the phage were now in 2 different places. All that remained was to ask where each radioactive label had gone. The answer was lopsided. The phosphorus, the DNA, was down in the pellet, inside the cells. The sulfur, the protein, was up in the liquid, left outside. The blender had knocked about 3/4 of it loose, while most of the phosphorus stayed with the cells. It was not perfectly clean. A little protein clung on, a little DNA washed away, but the trend was unmistakable. And the clincher, when those cells burst into a new generation, the young phage carried 1/3 of the original DNA and less than 1% of the original protein. The coat stayed at the door. The DNA went in and was passed on. So the thing a phage injects to build its next generation is its DNA, not its protein. Hershey and Chase were careful about it. Their paper claimed only that the protein probably has no function inside the cell, and that the DNA has some function, and warned against reading any more into it. But it pointed hard in one direction, and it agreed with Avery. Coming from the influential phage group in a single clean picture, it swung the field to DNA, part of the work that would later bring Hershey a share of the Nobel Prize. Its beauty is its thrift. No new machine, no grand theory. A borrowed virus, 2 radioactive atoms, and an appliance you could buy in a shop. Hershey and Chase never tried to out-argue the people who loved protein. They arranged the world so the answer would sort itself into 2 tubes. One you keep, one you pour away. A question with 2 possible answers, built to answer itself. A year later, Watson and Crick drew the double helix, and the molecule everyone had underestimated became the most famous in biology. Sometimes the favorite is wrong, and it takes a blender to prove it. Beautiful experiments.